the next talks, and the rest of our speakers are, in fact, here. So with Anish, you have no possibility to ask him, maybe by email, if you have further questions, but with the rest of the speakers, it should be easier. So our next speaker will be the Yasser El-Manzalawy from the Pennsylvania State University. And he's going to talk about the multi-omics data integration, so the cancer data integration challenge. Yeah, there should be a presentation in the...
Hi everyone, my name is Yasser El-Manzalawy and I'm from Penn State. Basically, I am an applied machine learning researcher and I have some experience applying machine learning to develop methods and tools for computational immunology and structural bioinformatics. And recently, I made a transition from bioinformatics to translational bioinformatics and microbiome research. And this is my first time I participate in CAMDA. And I'm here for the next 10 minutes to share with you some lessons that I learned from my participation. So I'm sorry, the word transition is just because the slides have been prepared with Keynote, not Microsoft PowerPoint. But as you might already know that advances in next generation sequencing technology and also large-scale efforts like the TCGA project has enabled the creation of huge and massive amount of multi-omics data. And they left us with the challenge of how to combine, how to combine all these different data sources to build a more reliable predictive model for predicting the clinical outcome. And simply, the problem definition is that if we have a set of patients, and for this set of patients we have different sources of omics data, our goal is how to integrate these different multiple data sources, or we call them multiple views, to build a classifier to discriminate between two phenotypes. And baseline approach is just we can integrate these multiple data sources into a single view and train a classifier, and this is called data fusion. And the other approach is that we can train a classifier separately from each data source, and then we just aggregate the prediction from all the classifier, and this is called the multi-classifier fusion. And this basic approach has limitation, and advances in machine learning research now have proposed something called multi-view learning. And in multi-view learning, we can train the classifier jointly, such that we end up with a better predictive performance than training separate classifier. And the same apply to do multi-view feature selection instead of just selecting feature from each view separately. Let me just show you an example of a multi-view feature selection. So canonical correlation analysis, this is an old and classically widely used data statistical analysis approach. And the idea, if we have two views of the data, our goal is to learn two projection matrices to project the input data into a reduced dimensionality such that they are highly linearly correlated in the reduced dimension. And this can be viewed as a multi-view dimensionality reduction. And what we did is that we proposed to use the learned matrices to score the input features, and by doing this, we are now doing multi-view feature selection. Another multi-view feature selection algorithm that we developed was adapted from the widely used algorithm MRMR. And the idea for MRMR is it's widely applied to gene expression data, is to identify the minimal subset of features that is relevant for the classification task. And basically, this is based on a function f that measures how relevant is the feature for predicting the class label, and another function g that determines the redundancy between two features. And adapting this for doing multi-view can be shown in here. Assume we have two views. So first, we will apply the function f to every feature to assign it a score of how relevant is the feature for predicting the class label. And then we will pick the feature with the highest score, add it to the set of selected feature. And then we just toss a coin, and this coin could be unbiased coin or biased based on domain knowledge to decide on one of the views. Once we decide on one of the views, let's say it is the green one, then we can rescore the features in here based on how relevant this feature for predicting the label Y, and also based on the redundancy with whatever we have in S. So we'll end up with different score for the features, pick the one with the highest score, add it to S, and we keep iteratively doing this until we add key features. So what we also did, when we heard about the CAMDA challenges, so they challenged us to find one solution that works for two data sets. And one of these data sets is the breast cancer. And for the breast cancer, they exposed us with two views, one from the microarray and the other one from the copy number alternation. And the neuroblastoma data set has microarray and RNA-seq, and RNA-seq data can be manipulated using 3 different software pipeline to generate 3 representations. So basically, we have 4 views for the neuroblastoma and 2 views to integrate for the breast cancer data set. And we have this data integration framework. So it has 2 stages. At the first stage, we can apply just a traditional feature selection method. So it will be applied to every view individually. And the other one can be a traditional feature selection. And in that case, it will be applied to the concatenation of the input. And if it is a multi-view feature selection method, then it will do multi-view feature selection. And this framework can be customized in different ways. So we can disable the signal for some features. So now we do the training based on view 1 only. So this is a single view predictions. Or we can just add a specific feature here that do nothing. So we will just bypass the input to here, and then we can use a traditional feature selection. So it will be applied to the concatenation of the input. Or we can do feature selection here and do nothing here, so we will concatenate the selected feature. And finally, we can do multi-view feature selection. We also experimented with a basic approach of doing multiple classifier fusion. So we can have a layer here where we can do feature selection, and then we will train classifier separately, and then we just aggregate the prediction. So at the end, we are comparing multiple classifier fusion with data fusion using these 2 multi-view approaches and concatenation of input or concatenation of selected feature, and also different ways of doing multi-classifier fusion. But in this talk, I will just focus on aggregating the prediction using the average of the predicted score. So this is the result for the MetaBreak dataset. So, and this is using random forest, and this is using another classifier called the extreme gradient boost. So this is an improved implementation of the traditional AdaBoost algorithm. And both classifiers agrees with each other that we can reach an acceptable performance using the macroarray, but we cannot reach a good performance using the copy number variation. And now it is not surprising that applying the multiple classifier fusion will not help. But doing data fusion using our multi-view feature selection slightly improve over the baseline approaches of just concatenating the feature, the input, or concatenating the selected feature. The story is different with the neuroblastoma dataset. So clearly, so let's just focus on random forest. All the views can lead to a good classifier. And the best classifier can be obtained using the magic view representation of the data and using only 10 features. And now it is not surprising that probably just fusing these 4 good models might lead to improvement. And that was the case. So we can have 1% improvement using just 30 features. So if using this 4 classifier, we have improvement, and you can reach the improvement using actually using more features. But surprisingly, the multi-view feature selection does not work. So what we learned from here is that, or before I move to this, I guess, like if I have to select one model from all these models that we have played with, I will select the single view one based on the magic view pipeline. And this is the 10 features that have been used. And 4 out of the 10 features are non-coding genes. So we also looked at the CAMDA 2017 because the neuroblastoma dataset has been used, and we found some evidence that they also reached the same conclusion. So basically, the integration marginally improved the predictions, and they also tried to use the copy number variation, which is available only for 145 patients, and it helped for some classification tasks, but it didn't help for predicting the survival, and this is something to explore in the future. But the problem with our method is that we expect that for every subject that we include in our study, we have all the views. So in order to apply our approach to the copy number variation data, we need to have copy number variation for all the subjects. And if you look at, this is just a typical data set extracted from the TCGA, and we have 3 views, and basically we have the phenotype for more than 900 subjects. But to enforce these complete views, we will reduce the data from more than 900 to only 300. So there is an urgent need to develop multi-view feature selection method that can be applied to incomplete views data. And in conclusion, we have shown that there is no one-size-fits-all solution to this challenge. And actually, if there is a solution that works for the 2 data sets, probably we can do better on every data set separately by exploring some unique characteristic of this data. And starting by just evaluating the single view classifier is a necessary and important step, and it can guide us to decide whether to apply data fusion or multiple classifier fusion. So if we have difficulty developing more than one classifier with acceptable performance, then there is no point of trying multiple classifier fusion, and we should do data fusion. And if you decided to go with the data fusion, then multi-view feature selection is one thing that you should consider. It has promising results on our published work on other data sets from ovarian cancer and kidney cancer. And if you can have more than one classifier with acceptable performance, then probably you should go with the multi-classifier fusion. Thank you. I'm sorry if I ran out of time.
Thank you, Yasser, for